So to maybe start, I would like to introduce myself. I'm Helene Steiner, and unlike the most in the room, I'm actually a designer and I'm an engineer. And I have my master's in design and science from Imperial College in London and the Royal College of Art in innovation design engineering, but it's about interdisciplinary work. And I had as well the pleasure to collaborate with the Tangible Media Group at the MIT Media Lab and MIT Chemical Engineering. What really opened up a whole new world of including life sciences into my practice. While my time as a researcher at Microsoft Research enabled me to get much better insights in the potential of computation. So as a result for me, really design, engineering and science started to naturally merge in my way of thinking. What makes me really excited to discuss with you today, today's session, Biointerfaces, blurring the borders between biological and digital computation. As a designer and engineer, I come from a different perspective to this topic, and I will use the terminologies interface and computation within a wider framework during this session. So my definition, my personal definition of a biointerface is the combination of biological and digital computation to create novel interfaces, which I would like to illustrate in a moment on two examples. And I will use the term interface in terms of applications where two systems come together and have an effect on each other. In my case, I will extend the definition, which is up here, and not only use it as a connection between a computer or a computer and a person. I will also use it in terms of an interface between a computer and another organism and a person and another organism. And further, I will use it in terms of communicating between disciplines and their systems as an enabling tool. The term of computation will be used as a metaphor during my talk for processing information to create a desired or calculated output. I will use it for biological and digital processes and the combination of both, which might lead into one of our discussion points in the end of the panel, the potential need of new terminologies in the field of bioengineering and interfaces, and the effects and limitations of using computational metaphors for the engineering of biology. All projects I will show today illustrate the need of multiple disciplines and the different perspectives and methods, which I think fits beautifully in this ethos of this conference. This relationship between the discipline is very beautifully illustrated by Professor Mary Oxman from MIT in her Grape Cycle of Creativity. Professor Oxman names her own goal to establish a tentative yet holistic cartography of the interrelation between the different domains, where one realm can incite revolution and evolution inside another, and where a single individual or project can reside in multiple dominions. The output, creative energy, not unlike the output of chemical energy in living cells, resulting from the fluid movement from one realm to another. The first example of such a project and a biointerface I want to give is Project Florence. It focuses on creating an interface between a plant and a person by understanding the processes within a plant cell and human language. During my time at Microsoft Research, we got interested in how living plants function and got fascinated that plants are constantly communicating with the environment and within themselves. And I would like to also say, you can picture our experiments. They're not happening in a biology lab. They happened in an office, in a design studio, and in an electronics lab. So they look a little bit different. And what I found so fascinating that because they can't move and therefore they have to constantly adapt and react to the surrounding to be actually able to survive. And one way how they are doing this is for electrical chemical signals, and electrical signals is something that we are actually quite familiar with, or most of us. So we began to capture those tiny signals from the surface of a plant with the help of a relatively simple tool we engineered that is not too far away from how we actually capture our own biosignals. And the device we built is less sophisticated, but from its principle, close to how the action potentials from the brain get captured, as we heard today in the morning. So we started as well to look into the patterns generated. And the fascinating part is that the electrical patterns are distinct to different abiotic and biotic stress, which provides the opportunity to map these patterns to different environmental causes and to find a way to be able to translate and as well maybe understand them in the future. It would represent a first basic interface between a plant and a person. Here an example of the signal of triggering and closing of a Venus flytrap captured with our current device. So you can see, or you just saw, how we twice triggered this tiny little hair inside the trap and the signal that it comes out of it and the closing of the trap itself. And we repeated this experiment very often, and the pattern is actually pretty close to similar. So to extend this concept of an interface into a two-way communication, you can as well recreate this motion of the fly trap by applying this electrical signal back to the plant, originally shown by Volkov et al. In his publication, Closing of Venus Flytrap by Electrical Stimulation of Motor Cells. And with Project Florence, we wanted to explore this way of a two-way communication between a plant and a human further. And I will show you now a quick video and explain it afterwards. To always spin the story a little bit further, what is possible. But in this project, we used language as a human input and light frequencies as a plant input, and translated for the help of natural language programming these human sentences in different light frequencies and durations. So it's quite similar to a Morse code, but sent through a biological organism, and then translated and captured the signals from the plant back and translated it back into human sentences, what you saw there. And here, for example, how our experimental setup really looked like. So here we control actually artificial light, like the intensity, the color, and the duration provided to a plant over the period of a week. And the output of the experiment shows changes of the variation potentials and fluorescence emissions in the plant in correlation to the light provided. So we blocked out any other light actually in the room while the experiment. And it looks quite similar to a Morse code, I would say, and I really hope that the plant was not actually complaining about me. But this project was for us really kind of a first step of exploring to turn on chemical reactions in a plant digitally. And even if this approach might seem a little bit of abstract, sometimes I think an unusual perspective can open up new opportunities. The second example I would like to share today is Project Biologic, which encouraged me to bring biology actually into my work. And Project Biologic was in collaboration with the MIT Media Lab and MIT Chemical Engineering. It is a growing living actuator and a responsive bioskin. The application you just saw is a garment that reacts to the wearer's body, temperature and sweat, and supports the body through controlling these tiny little flaps that you saw by opening and closing regarding the wearer's need. So for example, if you imagine that you are going for a run and in the beginning your body is still cooled down, so you want actually that your clothes protects you and keeps the body temperature. But when your muscles warm up and you start sweating, you want to transparent, what our skin is naturally doing. But our clothes is actually at the moment not really supporting this function. The idea is here that that garment actually supports the wearer's need. And when you cool down, all these tiny flaps actually start closing again. And the technology that enables this are actually bacteria cells. They have an incredible ability to expand and contract regarding to the relative humidity surrounding it. And through applying those bacteria cells on a substrate material, we were actually able to create shape-changing materials. And those cells, called Bacillus subtilis, are actually edible and are present in fermented soybeans for the Japanese food lovers. It's called natto, what you just saw, but can be as well found in soil for the ones who prefer digging in the dirt, but are also very common in many research labs actually. And the beauty is that those bacteria cells, or bacteria cells in general, can duplicate themselves billions of times, and the exponential growth rate enables the use of such growing actors actually on a big scale. But I think the really exciting part, for me at least, is the sensitivity to its environment. It opens up new biological interfaces without the need of embedding any electronics in comparison to Project Florence. So we used our own skin as the interface that creates a direct interaction between the human body and bacteria cells. And in Project Biologic, we needed computation as an enabling tool to build the interface. We built a software which enabled us to really engineer the fully functional garment by combining the data of the female and male sweat and heat map in combination with the data we collected from the bacteria cells of its expansion rate regarding to the relative humidity, and in combination of its reaction to the substrate used. So the software creates a simulation and feedback loop of the interaction and material we actually design. And connected with a hardware platform, in our case a bioprinter, we were able to produce our idea one-to-one and test it out, and as well make it accessible for other designers, engineers, and scientists, which enabled us to create a wider range and ideas of other biointerfaces and applications, from shape-changing lamps to animated origami to artificial plants. And very recently, just in the end of last year actually, we published in Science Advances an extension of the capability of such interface, which now incorporates as well light emission alongside shape changes. So here we used a similar approach I just showed and used green fluorescent proteins expressed in E. coli and the reaction to the relative humidity as well. So our example was actually a shoe that changes its color and opens up and close for nut smelly feet. But all the projects that I just showed, or both projects I showed, are really only possible because of a big interdisciplinary team. And it was really only possible because of these different mindsets, skill sets, and really bringing different fields together to have a conversation and bringing something to reality. And the projects just shown showcase designed interfaces between a person and another organism. One between a plant and a person, the other one between bacteria cells and a person. And both were created based on the capability of living organisms to react to different stimuli in its environment, and are used as a function or calculated input-output system. Particularly in bioengineering, makes advantage of using these engineering principles and terminologies from computation. In 1996, Tom Knight convened a series of meetings on cellular computing, launching the field of synthetic biology. These early conversations fleshed out a set of valuable computational metaphors for use in the engineering of biology, such as living sensors and actuators, logic gates and switches, DNA as software and proteins as hardware for future biocomputational interfaces. Today, these converging metaphors are shaping increasingly powerful technologies that are influencing so many aspects of contemporary life, and perhaps even life itself. And Christina Akapakis from Ginkgo Bioworks would have actually particularly given us more insights about this, her criticism, her practice, and also the potential idea of replacing technology with biology. And this approach, in combination with computation, allows us to deal with an otherwise impossible complexity. Biology in itself is already very complex, and even more in combination with other systems. For those familiar with engineering, computer-aided design tools, or called CAD, they are fundamental to design and engineer ideas by using computer systems which aid the creation, modification, analysis, or optimization of complex designs. And software helps to increase not only productivity, but also to improve the quality and precision, improves communication through documentation, and to create a database, all which is needed to apply biology. So both projects wouldn't have been possible without such computational power and aid. And they're used to process data and act as a conductor or translator to enable us to understand processes within other organisms, to be able to engage with them, and acted as an enabling tool to give us the opportunity to engineer and design new and powerful interactions, which otherwise wouldn't be possible because of the complexity behind. To bring the design out of the test tube and the laboratory, hardware is needed as the other enabling technology for biointerfaces, which allows to make it tangible. This is only possible because of advances in manufacturing methods, such as 3D printing and material sciences in the last years. The capability to print microchannels to interact with single cells in form of microfluidics, printing moving textiles made of bacteria cells like you just saw in Biologic, or weaving ties made of bioengineered spider silks like Bolt Thread is started to do now, wouldn't have been possible a few years ago. Hardware allows to engage with biological materials in 3D space, to study, engage, manipulate, or combine them with other materials. And open lab and hardware tools, such as used in Florence, are here a big enabler for the whole community. So David will give us in a moment a much more in-depth overview about the potential of such enabling technologies in non-traditional and creative ecosystems, including open tools, virtual infrastructure for sharing, new programs for learning and education, and mechanisms for delivering cultural impact. We're talking about the need of hardware. At the company I'm working now, we are developing open hardware for biological sciences and cross-disciplinary curriculums. The goal is to build tangible representations of otherwise difficult biological processes. Our goal was to produce this for under $100 to empower labs and communities all over the world. We call it Pixels, and it's an educational tool which allows students to build their own biological display by using fluorescent proteins. So what you can see here is the full kit, and it includes electronics and a digital interface allowing students to build a low-cost fluorescent protein visualizer. In the lab also called a plate reader or a transilluminator, while learning about hardware and programming. So inside the gray box there's actually an open source electronic kit. And it also has exchangeable optical filters and a cover to learn about optics and integrate physics into the curriculum. And as well a pipette, the most important tool for biologists, and cell-free extract and DNA to be able to produce your own fluorescent proteins. And it is built for letting students modify and play with it and be really flexible. I mostly use it actually for playing Biotetris. So that is actually really built with the little kit. So you can program it to go beyond just a plate reader. And the key part of Pixels are actually the biological parts. Transforming or getting DNA into cells cannot be easily done outside the lab because of the danger of reproduction and the tools and time required. To use the cell is a lot like a factory. At least that's how I see it. That's my representation of a cell. The part responsible for manufacturing, what you see kind of like in the middle here, takes up only 5% of the cell, and its job is to read DNA and write proteins. We work with cell-free systems, meaning we actually break the cells open and work only with the protein-producing translation transcription machinery outside the cell, which allows us to prototype and produce proteins in a fraction of the time and outside the lab. So we don't know much yet how living organisms really function. Biology is about guessing, prototyping, testing, and back to guessing. What you see here is our first approach of a design tool to help this process and to let a wider variety of people taking part in it. Our platform, Biobits, allows to submit your DNA sequence online. This sequence gets tested out in our laboratory in one of our cell-free systems that I just mentioned, and we send back a report within just a few hours of the performance of the DNA sequence, such as the intensity of the expression of a green fluorescent protein. The database is created open source, because looking at the challenges that lie ahead, we think they can only be solved by a collaboration of minds and vocations and a diversity of views. So if you are interested in sharing your ideas, please join us or talk to us later. And if you look back in time, disciplines were fairly siloed. Collaborations were still not very practiced, and interaction between fields not really seamless. Today, the walls between disciplines are blurring. New fields started to emerge out of this and enabled many innovations. I believe biointerfaces can be the next frontier towards a more intuitive, intelligent, hybrid practice that reaches across the disciplines and fields to create seamless, sustainable, beautiful, and intelligent applications and communities that will take us in the next century. I'm personally excited to see where it goes and to hear your insights and perspectives later in our discussion. And somehow the text on that slide I just see actually got lost. So maybe we can fill in all these future fields that can emerge out together. But first, now I'm excited to actually welcome Dr. David Kong, from the Director of the Community Biotechnology Initiative at MIT Media Lab. Welcome.